our infectious disease pandemic expert. So, Bob, it's great to see you again, and I look forward to what I hope will be a very lively next 50 minutes. Just a few housekeeping issues as we get started. Please use your chat on the bottom to ask questions or the question icon. I'll be checking both of them throughout the session. Well, before we begin with our update on vaccines, which are clearly everybody's highest interest right now, what I thought would be nice is for us to review where we are right now, both in the state and at university hospitals. And, you know, we like to give you the state of Ohio COVID statistics. We have our red light, green light, yellow light, and you see that yellow light flashing. And we'll go through this just very quickly. You definitely see in the last 24 hours a downtrend in both cases, deaths, hospitalizations, and also importantly in ICU admissions, suggesting that there is a decrease in disease burden right now across Ohio. However, I think you'll see in the 21-day trend diagrams that there's a lot of spiking going on. So we're going up, we're going down. Cases have shifted by about 15 years in median age to lower aged individuals, with obviously a big bolus of cases, as we all know, amongst our students attending universities in Dayton, Miami of Ohio, Ohio State, and other areas that are causing increased caseload and potential exposure to higher risk individuals. The good news, though, as you can see here in the next slide, which is entitled University Hospitals Health System COVID Cases, that on today, September 20th, we are really at a good low across the health system with only 21 cases across our 12 medical centers. And you can see that's about as low a number as we've had since all the way back in the third week of March. So a nice downtrend in Northeast Ohio as well. Well, what I'd like to do now, Bob, as we get ready to talk about vaccines, is to just spend a few minutes, if we could, on what we call vaccines for dummies, or vaccine biology made easy. And so what I'd like to do is just spend a few secs going through a couple of slides about the biology, and then you and I will dive in into some further question and answers. Well, the first thing I'd like to do is just remind the audience to focus on those red triangles or spike proteins that you see on this model of a coronavirus, because that's really going to be the basis of protection of our vaccines. And we'll get to that in just a moment. Now, unfortunately, I got to bring everybody back to high school biology. And there will be a test on this material, Enid Rosenberg, so pay attention as you're listening now. This is the central dogma of biology. So if you go all the way back to whatever your sophomore year in high school, you remember that every cell has a nucleus, and inside that nucleus, there's DNA, which is our genetic code. The DNA can replicate in a process called transcription. It makes something called RNA, and then RNA is translated into protein. And the reason why this is important is because in vaccine technology, we are going to have DNA vaccines, RNA vaccines, and even protein vaccines. And we want you all to be able to understand some of those differences. Now, if you go to this next slide, which has really the 4 types of vaccines that we have available, there are genetic vaccines, which are DNA or RNA, and some good examples of those, and we're going to talk about them in some detail, are the RNA vaccines of Pfizer that we're undergoing testing now at university hospitals, and the RNA vaccines of Moderna that you've heard about a Cambridge-based biotech company in the US. There are viral vector vaccines, and the most notable of that is the Oxford AstraZeneca vaccine, and that viral vector is a monkey adenovirus, which does not replicate in humans, but which is used to bring the information that will code for that spike protein. Of course, there are also protein-based vaccines, which are the kind of vaccines that many of you are familiar with like the flu shots that we get. And then, although not presently in trial at university hospitals or in the United States, there are actually vaccines related to inactivated or attenuated coronaviruses. So with that, I'm going to stop sharing, and I'm going to ask Bob, I think, the question that everybody wants to know, and then we'll really start diving into this. And that is, tell us a little bit about, very high level, where are we in vaccines right now, and what is the prospect for vaccine rollout in the US? That's a question that we'll spend a lot of time on, but is obviously something that everybody wants to know right out of the gate.
Sure. So it's been an amazing trip to get to this point in time where we're testing these vaccines in the last stages, the so-called phase 3. And I know you have a slide later, Dan, where you're going to demonstrate that. But this is not only continuing to look at safety, but now efficacy, that is how effective these vaccines are. And we're looking at several parameters to both surrogate and primary parameters to assess efficacy. One of which is the development of antibodies, and you're going to cover that in a slide. These are so-called neutralizing antibodies, which inactivate, if you will, the spike protein. And that can be measured in the laboratory, including, and most importantly, in these individuals receiving the vaccine or placebo, and we make the comparison. The other is so-called T-cell responses. This is another form of immunity that is also important because they're longer acting, and they may provide additional protection and will uncover whether or not that in and of itself is sufficient to explain the protection for vaccinated individuals. So those are very important immunologic matters that we're looking at. Of course, the primary objective in all of these phase 3 trials is to look at the occurrence of COVID infection in the vaccinees versus those that receive placebo or control, same applies. And in that regard, that's why we're going to be following individuals for a longer period of time with respect to that parameter. However, in terms of manufacturing, most of these companies are hedging their bets and are manufacturing vaccine now, including quite a bit of it. And the promise, for instance, for the vaccine that we're involved with, where I'm the principal investigator for Pfizer, coupled with a company in Germany called BioNTech, is that they were given $1.95 billion by the feds in the US to deliver 100 million vaccines by the end of December. In terms of our vaccine study, and this applies to the others as well, there are 3 of them that are active in the US right now. We are well on our way to completing our first round of vaccinations in individuals. Well over half of our target of 250 have been vaccinated to date. And we plan to finish up the vaccines in the first round by at least the first week in October. Following that, everyone will receive a second vaccine. That's an important concept here in terms of the total number of available vaccines we're going to have early on, in that that is what it takes to have the most robust or maximal immune response. The data regarding Pfizer's vaccine, for instance, will be reviewed by our Food and Drug Administration, FDA, and others. And there's an external group that will look at this very carefully. And as you probably have heard, all of the major manufacturers of vaccines have submitted a pledge not to release their vaccines until they're really sure about both safety and efficacy. And in that regard, I don't think we're going to have vaccine available by the election, as has been said from a political point of view. I think the best we can expect is that by the end of December, to have some vaccine available. And we could talk about the priorities for that, because there's been a Human Health Services playbook actually published recently in terms of who should get it first. And it will take at least 6 months thereafter to have sufficient vaccine to offer to individuals in the United States, given our overall population. We can also talk later, Dan, about polls indicating the numbers of folks that would be interested in receiving vaccine at this time, those that are uncertain and those that will not. But that's where we're at right now.
Great. Well, you alluded to this concept, Bob, of, and we should review this for a moment. Exactly how does a vaccine work? And you can see in this diagram that there's an antibody at the top that's produced in response to a vaccine, and it binds to those spike proteins. You could think about this a little bit like getting the mail delivered to your mailbox. The mail is the virus. It's got to find your mailbox. And let's just say your address and your zip code is your address. And so this virus uses a receptor called the ACE2 receptor to get into cells. And this antibody basically binds to those spike proteins and prevents them from entering the cells. Now, I think, Bob, I'm going to let you explain this slide, which is really very, very important, because it gives off concepts related to the different phases and where we are right now. At least I know there are over 100 different vaccine candidates, but these are certainly in the coronavirus vaccine tracker that's being published regularly by the New York Times below. This is the latest in phase 1 and phase 3 trials. So maybe you could go through this and walk the audience through the different phases
Sure.
and what they're supposed to accomplish.
As you have pointed out, I think the number is about 120, the number of candidate vaccines being tested around the world. But all of them usually go through the initial steps, which is called phase 1. These are otherwise normal individuals, generally between the ages of 18 and 45, let's say. And the major objective here is to make sure the vaccine administered is safe. And but in the cases of the vaccine administered for coronavirus, they've also taken the opportunity to look at how well it produces in those healthy individuals the antibodies and T-cell responses that we spoke about before. Once that is accomplished, then they're allowed to move to phase 2, which in both of these cases, phase 1 and phase 2, these are smaller numbers of individuals with the objective that we already said. Phase 2 begins to, in many cases, enroll individuals who have some risk factors for the development of serious conditions related to COVID and death. And in fact, they looked at folks up to the age of 65 in many of these COVID vaccine trials in the phase 2 situation, primarily focused again on safety, but gaining that antibody and T-cell response data as well. When you pass those steps, you go to phase 3. And that's where we're at with the Moderna vaccine, the AstraZeneca vaccine, and the Pfizer vaccine that Dan mentioned already. These vaccines are generally larger, and in the case where we need important information in a rapid fashion, but not cutting any corners, these vaccines in all of these circumstances are enrolling anywhere from 30,000 to almost 60,000. One of the vaccines about to come about, which is protein-based, it's called Novavax. So right now there are limited vaccines that are being utilized. None of them have been approved at this point in time. As I said, there's this pledge that these manufacturers, these pharmaceutical companies will not release vaccine until we're certain about safety and efficacy. And we will move to approve something in accordance with that pathway. This is, as I mentioned before, something at the federal level called Operation Warp Speed. I wish they didn't use that term because it suggests maybe cutting corners, but I can tell you, being involved in this process, there are no corners being cut, that this has been taken very seriously. We have daily calls with Pfizer. In our circumstance, we review the safety data, and in the Pfizer vaccine trial, there have been no major safety signals so far.
So I guess couple questions are coming through, Bob, and maybe we should start to tackle them as we unravel this vaccine story. And the first question is a great one, and that is, what's the time between the first dose and the booster? In shingles, it's get the second dose by 6 months. The time here is shorter. Do you want to explain that?
Yeah, that's 3 weeks in our case. And the other vaccine trials, it could be as long as 4 weeks, but generally that's the time period. And that was assessed during those earlier stages of study that I mentioned as to when we saw maximal antibody responses in these individuals. So we're doing a 3-week period. The point also is this needs to be very carefully considered in terms of if we're going to roll out vaccine and when, we spoke to that a little bit, people will have to come back for a second injection. And the logistics around that are not trivial, as well as the fact that right now, let me just end with this, it is the case that this vaccine is stored in very, very cold temperatures. And you have to respect that issue. And this is probably not going to be such right now, although they're looking at different temperature ranges for stability and storage that we're going to offer this to many private physicians' offices, because the storage issues are such that this will probably need to be centralized. And so the logistics around putting up vaccines is really important and may delay, appropriately so, when we start rolling this out.
So Bob, there's some questions coming in about efficacy, and we certainly have heard in a lot of the phase 2 trials that you've talked about that the antibody titer of the vaccine is exceeding that of natural infection, applying potentially immunity. And then, of course, there are the T cell responses. One of the questions that we've received is this issue of how efficacious does a vaccine have to be? I mean, we know that the flu vaccine, which does reduce the number of flu cases, is only about 40 to 60%, efficacious. But with flu, one, the disease is milder, and two, there's Tamiflu. We have a reasonable antiviral agent as well. So maybe using that analogy, what do you think the efficacy of the Moderna, Pfizer, and AstraZeneca vaccines is going to look like? Certainly, there's very exciting animal data in monkeys that it was almost 100% protective. But do you want to comment on that?
Sure, that's a great question. And the figure that's been pushed forth into the lay press has been anywhere from the 50% level that it's been indicated on the slide and questioned here, and up to 70%. One of my dear colleagues, and probably the foremost expert in the country for infectious diseases, Dr. Anthony Fauci, has said that if we get up to a level of 70%, we'll really have achieved something phenomenal. But even at 50%, and if this raises the so-called herd immunity levels, that is efficacy of antibody levels in the entire population, this really should make a major dent in terms of the pandemic and numbers. And the level for herd immunity, that is how many people are positive for the antibodies at different age groups across the population, is around as low as 43% and as high as 60% for us to really make a dent in this problem. So I think that's the first thing. And if we achieve this, somebody asked in turn, if I may, Dan, what efficacy means in that same question. So we're looking at the production of these antibodies that in the laboratory in turn neutralize the spike protein, and therefore if we take that further, as you already explained, will prevent the virus from ever attaching, even if exposed, in the respiratory tract to these cells, and then stopping or aborting the infection to begin with. And then that's an important figure in terms of just generating the antibodies. The T cell responses, we are less certain about at this time, but in other viral infections, T cell responses are also important and are also long lasting. And I think that's important for us to think about in terms of if we ever have to think about giving this vaccine again, as we do with influenza, and so on and so forth. So that's where we're at right now in terms of defining efficacy. And I mentioned before, the true measure of efficacy, effectiveness, will be to really look at the numbers of individuals being vaccinated versus those that get placebo in terms of true cases, documented cases of COVID-19. And if we could show that that's greatly ameliorated or decreased, I think that's but that's going to take some time. So we're going to start with these surrogate markers first. That is antibodies and T cell responses in conjunction with the safety measures to probably initiate vaccines while we're waiting for more information.
So a lot of questions, Bob, clearly about safety, given the signals in the AstraZeneca Oxford trial related to a rare complication called transverse myelitis, which is inflammation of the spinal cord and can be associated with temporary or sometimes longer lasting paralysis, weakness, and pain. And that's obviously a very rare complication associated with viral illness and with other vaccines. But this really raises, I think, a very important question. Even in a trial that has 30,000, 40,000, 50,000, if you're looking at something which is very, very rare, single cases become very important. And we all remember back to the swine flu example, when there was about a 10 to 50-fold increase in Guillain-Barré, when very small numbers of Guillain-Barré cases really came forward and put a little bit of a break on that vaccination program. So tell us what you know about the AstraZeneca program. It was restarted in Europe. It has not restarted in the US. Should we be concerned about the case of transverse myelitis, and what do you think is going to happen?
That's a great question. And obviously, this has to be vetted very, very carefully. I might start by mentioning, Dan, that my own son, youngest son, developed a form of Guillain-Barré after receiving a flu vaccine as an example. So it's hit home personally for me. Yes. But he recovered totally and is doing well, but he'll never get a flu vaccine again. With that said, this is a rare event. And as Dan said, this can be associated transverse myelitis, inflammation of the spinal cord, depending on the level of the spinal cord involved, with paralysis of usually the legs, sometimes the arms as well, and also pain, etc. In this case, there were two instances of transverse myelitis seen in the AstraZeneca program. One was in the early stages, which has not been well publicized. And to that end, it was felt they had very good evidence to suggest that individual developed multiple sclerosis as the basis for this phenomena. And this can occur with multiple sclerosis. In the second case, during the phase 3 initiatives, this was a single case reported in early September, causing initially halting globally of their vaccine program. This was vetted by a data safety monitoring committee, which are all folks that are independent, highly expert in the area. And they decided to report to their equivalent of the FDA that they thought the vaccine should be restarted, at least in the UK initially. To that end, it was felt they could not firmly establish a cause and effect relationship. And that's part of the issue here. And given the rarity of this, I'm also led to believe through my information channels that this individual has had significant recovery, though it required high dose of steroids to bring this about. I took the opportunity because that question is being asked widely, and especially among our recipients of the Pfizer vaccine, to look at this more carefully, at least in the US. There's a database kept by our FDA of vaccine-associated serious side effects that have been reported on an annual basis. And in the last 15 years, there were 119 cases of transverse myelitis reported. Now, it's interesting that only 35% of them occurred in a time frame that you would more likely believe would be associated with the vaccine per se. And it's been a variety of vaccines as well. And whereas the majority of these cases, even though that represented 65% or so, were seen months and months and even years afterwards. And it's really hard to know whether the vaccine was the trigger or not. As you mentioned, there are a variety of other viral infections, autoimmune diseases, like multiple sclerosis or lupus that can be associated with this phenomena. And sometimes you just don't know the basis for it. Right now, the US has not decided to restart the vaccine program related to the AstraZeneca vaccine. There is independent review, not only by our FDA, but the NIH, because the NIH is partnering with AstraZeneca to push this vaccine out here in the US. But they are not allowing this program to start until we have that review and are satisfied that we cannot clearly associate it with the vaccine. But that's where things stand right now.
Okay, so Bob, bunch of questions really fast. One, any concerns about an RNA vaccine? Is it possible that the RNA could somehow influence your DNA either by getting incorporated or somehow even binding to DNA causing mutations? So concern about safety of RNA.
That's a very great but sophisticated question.
Yes.
Because this is genetic material that you're injecting linked to the spike protein so that the RNA, messenger RNA, in fact, that's why it's called mRNA, yep, is such that it's presenting the antigens to the rest of the immune system for the responses to occur. There have been concerns about mRNA being incorporated permanently into our genetic material. And this would just nonstop be present and even producing exorbitant amounts of the protein that it's linked to. But actually in preliminary studies, even before this was ever put into humans, it's been established that this is extinguished after this messenger RNA after several months yes, at most. And I think that is an important concept. Messenger RNA or mRNA-based vaccines have been used in other scenarios. So there's a track record of its safe use and established that it does not set off a permanent residency within our own genetic material. One of them is, for instance, the recently released Ebola vaccine made by Merck. And this is an mRNA-based vaccine, and it was effective not only in West Africa, but also now in the Congo in helping to really diminish disease. In fact, in countries like that border the Congo, like Uganda, they were very precocious in terms of setting forth a vaccine program in their healthcare workers on the border. And they had no cases there for what it's worth. But that's just another example of a messenger RNA-based vaccine. So the genetic DNA or RNA vaccines are becoming more used over time in other circumstances. The beauty of it is it's easy to produce, and you can make tons of it, if you will. And that's part of the manufacturing logistic challenges that I spoke about before.
Yep. So Bob, one of the questions, and we'll ask our development team to send it out. There are people who are still interested in volunteering for the Pfizer trial. Now, I understand that you're trying to enrich for higher risk groups, especially the elderly. We do enroll up to 85 in this trial, those with concomitant high risk medical conditions, and most importantly, a broader representation of females, African Americans, and Latino individuals. So we have the groups that have had higher disease burden broadly represented. So we will send out information. There are slots available still in the trial. And because Pfizer has increased the trial size from 30,000 to 44,000, we are likely to have more slots. You do not need by any means to be a UH employee to get into the vaccine trial. We're looking for all volunteers, and we will send out contact information to all of you in case you are interested in volunteering. I should say from my own personal experience, I was enrolled last Tuesday, Bob. It was a very, very thorough process. It is an extensive day of investigation. You are there for 2.5 hours for the first visit, because not only is there a very careful review of your medical history, but as Bob pointed out, the mRNA vaccine is cooled or frozen, so to speak, in liquid nitrogen level temperatures. So we had to wait 30 minutes to thaw the vaccine. And then I was asked to stay for 30 minutes after I received the vaccine or placebo to make sure that I didn't have any acute side effects. And aside from feeling like I had a shingles vaccine type feel for a couple days in my arm and a little aching, everything went well. And I just hope that I got the real thing instead of placebo. So now, Bob, there's a question about, well, let's say that vaccines get approved. Would you make a recommendation that somebody should have multiple vaccines from different manufacturers so that they have optimal protection, or is that not known yet?
Another great question. Thank you for that. So my opinion is that because of the sheer numbers, when you look at this globally, we're going to need multiple vaccine options in the end. And I think, however, based upon the fact that there's not going to be in the short run any comparative studies between these vaccine platforms, that staying with one makes the most sense, because that will be thoroughly studied, both in terms of safety, in terms of antibody responses and T-cell responses. We would not suggest moving between unless there was such a shortage of one of the vaccines that you had to get the other. But I think it's reasonable based upon how these are being studied. No comparative studies at the moment, those might come later, that you stay with the same vaccine platform.
Okay, so now I'm going to go back to the slides for the real tricky question, Bob, about who actually is going to get the vaccine first, so to speak. And so I'm going to blow this up now. I think you can all see this. And this is a little bit of a figure, so to speak, from the recent playbook that was released by the FDA and I think the CDC related to the COVID-19 vaccination program and a phased approach. And I call your attention to the left-hand panel of this figure when we're talking about a period of limited doses available. And that's sort of highlighted in that left-hand panel called phase 1. And I draw your attention down to the bottom left-hand side where it explains who actually is going to be getting the vaccine in phase 1. So let me read that because the print's a little bit small. And it said, phase 1A will be paid and unpaid persons serving in healthcare settings who have the potential for direct or indirect exposure to patients or infectious materials and are unable to work from home. So phase 1A, you can think of as first responders and frontline healthcare workers, either taking care of COVID patients, emergency room, intensive care units, areas of the hospital where there are these patients, or those who are indirectly exposed in that care path. Phase 1B will be all other essential healthcare workers. And then as we get into phase 1B, people at higher risk of severe COVID illnesses, including those individuals who are 65 years of age and older. And I think the reason why it's important to review this, because as you go down or down on the bottom of phase 1, phase 2, and phase 3, you can see that really the general population really doesn't start getting vaccinated until sort of the end phases of phase 2 and into phase 3. So you're really talking about critical populations in phase 2, those are those patients with diabetes, obesity, heart disease, lung disease, active cancer, etc. So I do think, it is worth noting, Bob, that when you really get down to the fact that there are about 330 million Americans, and we're talking about the time that it's going to take to really vaccinate broadly the entire population, I think we start to understand that while we could be vaccinating, as you said, 50 million individuals from those 100 million doses from Pfizer, even starting, let's say, in December or January, until we start getting into, if you start thinking about, can our families get together for birthday parties and events? Can there be weddings without distancing and masking? I think you start realizing, Bob, that this is like a Q3 2021 issue. So do you want to talk a little bit about that and these phases? Because obviously you're going to be playing a critical role in determining who's getting vaccine and what priority in Northeast Ohio.
Right. So it's not going to be that, Dan, and I completely agree with your thoughts, but it is also how people will come to get vaccine because of the storage issues and the logistics right now. So I think we need to understand that, although we're expecting some vaccine to be delivered to the federal government by the end of December, it's going to take time to really ramp up to where we have this. And if I may, let me just mention some of the polls recently regarding vaccine acceptability for coronavirus. Some of the polls show that maybe 50% of people at most will readily take the vaccine as soon as it's available to them. Another 25% are uncertain at this time. And I think I have found in our own study here, Dan, that we've had several people, about 6 so far, turn us down because of the AstraZeneca situation, even though this is a totally different vaccine platform. And they need to be reassured more about the safety. And also about 25% of people say, no way in hell. And some of those are the anti-vaxxers. And you could understand how they've used this single rare case of the AstraZeneca vaccine platform to really push their agenda as well. So there are discussions, although not solidified yet, of mandating or considering mandating vaccine for school-age children and others like healthcare workers as we move forward in this regard. And that is something that's generally not done with vaccines, although it's becoming more so, for instance, in the pediatric age groups for measles. And we mandate influenza vaccine for our healthcare workers in our system. And that's because not only are we protecting them and increasing their preism, if you will, at work and decreasing the absenteeism, but also keeping their households safe and our patients especially, because there has been transmission of influenza from healthcare workers to our sickest patients. So that is going to be something that's going to be an interesting dialogue as we move along.
So Bob, there are a lot of questions from people who are putting 2 and 2 together and are saying, well, if you get placebo in the vaccine trial, when do they give you the real shot? Yeah. So how are placebos handled in these trials?
So it's going to be made by each manufacturer, but the agreement for the Pfizer vaccine right now is once the initial set of data is fully analyzed, they will then consider whether to give active vaccine to those that receive placebo. The problem is we need sufficient time after placebo and vaccine administration, time out in terms of knowing whether or not this does truly decrease the occurrences of true COVID-19 infection. So that's going to be handled as a very delicate point of balance, but they have assured candidates that receive placebo that they'll be among the first to be offered true vaccine as well.
So another interesting question, Bob, about the flu vaccine. Someone's asking, how come we don't get a flu booster? Maybe the efficacy of flu would be increased if we got 2 flu shots. And someone said, should I go get a second flu shot?
It's a great question. It's asked all the time. So for one, we do give a series of vaccines to very young children because they've not been sufficiently exposed to flu during multiple seasons, and therefore they need their immune response to be more robust. Keep in mind, though, we have to give the flu vaccine right now on an annual basis because the flu virus itself can change either subtly or in a major way, that is the outside covering of the flu virus itself, so that previous vaccines and exposure may not be sufficient to protect you going forward. The best example of this was 2009. Most of you know about this time where we saw a pandemic flu during that season. And although that one is still with us, by the way. But now we have sufficient immune responses from before to cover us going forward. But given the fact that the flu virus can change in the ways that I said, we have to consider that. We are working on vaccines for flu that will be given less frequently or maybe even a single dose, but we're not there yet. I would use this opportunity to remind folks to really make sure they get their flu vaccines this year. And for the first time, for folks that are 65 years of age or older, we have a high dose flu vaccine that contains all 4 of the prevailing strains. In the past, it only had been 3. That was the trade-off. And the high dose vaccine does provide stronger immune responses, keeps people out of the hospital because they don't develop a severe disease and die less frequently. And I would use this to jump to the coronavirus vaccine, which we haven't mentioned in that respect. Even though we're trying to achieve antibody levels and T-cell responses at 50 to 70% or greater, it doesn't mean that it's an all or none phenomenon. If getting vaccinated, as in the flu vaccine situation, means that if you develop COVID through the vaccine, you may have milder disease, have less severe complications, and die less frequently. That in and of itself, in my view, is a success. And so we have to keep that in mind, and that's part of the flu story. So get your flu vaccine this year to prevent what we're anticipating potentially as a twindemic, that is both the flu and coronavirus happening at the same time. And also about 12% of people can have both at the same time as well. And certainly the symptoms are overlapping, and getting your flu vaccine protects you against that. Lastly, I'll mention in the Southern Hemisphere, and this has really been also in the lay press, that they had a pretty mild flu season. Remember, they're opposite from us in terms of their winter time, et cetera. And that really had an impact. And we think the measures we put into place to prevent coronavirus, COVID-19, really impacted on influenza vaccine at the same time. Masks, distancing, hand hygiene, and the like. So we hopefully will have a milder flu season this year, but we don't know that because there are a lot of people not doing those types of things to prevent coronavirus infection to begin with, and that will apply to preventing influenza as well.
Bob, I did get a little note, probably from one of your research nurses who's watching, that if you are interested in enrolling in the Pfizer trial and you have not had your flu vaccine, do not go and get it, because in the trial, you have to have your flu vaccine 30 days after your booster or your second shot. If you have your flu vaccine before, you are disqualified from the trial for now. So
for 14 days. So if you
yes.
got it early on, that's absolutely true.
Okay, so just be a little bit careful about that timing. But if you have any interest in the trial, we will send you that contact information, and you certainly can call up and give the details of your particular case. Well, Bob, we only have a few remaining minutes before we wrap up. And I thought it'd be a good time for you maybe to just give a high-level summary recommendation of what people should be doing right now. I mean, one of the things that we have been trying to emphasize here is that nothing has changed related to the problem that we're in right now. And that is that all the measures of masking, distancing, engineering controls, and hand washing still remain very important because we don't have a widespread vaccine or protection yet. So Bob, any general recommendations for everybody before we conclude?
So you're absolutely right, Dan. We should continue those measures. And even after the vaccine becomes initially available, then more widespread in its availability, we're going to have to keep in mind that it's going to take time for that to work, if it does, to the extent that we expect. And therefore, I think we're in for the same approaches, at least through the summertime of 2021 and maybe even beyond that, before we make a big dent in this. And we hope, fervently, based upon everything we know and the animal data that you also discussed, Dan, that these vaccines will be effective and we'll be able to make a big dent. And this was articulated recently by the CDC as well, that just with upcoming vaccines, we can't back off from these issues. But that remains a problem in the US. People are not necessarily adhering to these. And the anti-maskers and the anti-vaxxers are getting together now. So, they consider it an impingement upon their own individual freedom and so on and so forth. But, we're in the middle of a worldwide pandemic. In the US, we're approaching, maybe today we'll surpass 200,000 deaths. And it is a real problem. We're approaching 10 million deaths worldwide as well. US still leads the list among the frequency of infections and death rates seen globally. We're closely followed by India, and given their populations, it may be the fact that in several months they'll surpass us. Brazil is third. So we have to undertake these same measures and take this seriously. One of the major points of education for us is in terms of the epidemic, as you've already described, Dan, with folks 1.5 decades lower in age that are getting this now. Although they're less at risk, especially in the context of folks enrolled in universities, et cetera, we're seeing spikes now. For severe complications, that is true. But as you mentioned, they can be the source of transmission to the aged and also folks with vulnerable conditions. So I think that's very important to keep in mind as we move through. And so far, the message has not gotten through to these young folks. When you continue to see large gatherings, no masks, et cetera, they think they're invulnerable. And I would just rightfully point out that this has to be a major thing. So for your teenage grandchildren and others, even in the elementary school age groups, we need to stress this. And school children can be the vectors for transmission. That is the source of transmission as well. And some recent studies from South Korea, which has been way ahead of the US in terms of trying to control this, have demonstrated that often school children who get this infection are less symptomatic or asymptomatic, but are spreading this to others. So I think as this pandemic has evolved, we've learned many things. We've never seen anything like this. It's unprecedented in my lifetime for sure. But I do firmly believe that in the end, the vaccine will make a difference based upon everything we know. But it's going to take some time for that to happen.
Well, I think, it's a good jump off point, Bob, to take that analogy back to flu. Yeah. And we need more than just a vaccine. We need antiviral therapy, which is either prophylactic. So you've heard about, for instance, in nursing home populations that have an outbreak, to give the new Lilly monoclonal antibody cocktail, which is effective at reducing infection, we believe, based on the first clinical trial. It's also generating a Tamiflu equivalent. So whether it's intranasal, remdesivir, or other drugs that kill virus. So we have more tools in the tool chest. We know that even if a vaccine is successful, we want antiviral therapies that can kill the virus. The good thing, and I want to remind all of our friends who are watching, is that University Hospitals is leading the way in research related to not only antiviral therapies, but also vaccines for the next pandemic. So the Harrington Discovery Institute launched a national-wide request for research proposals, received over 250 proposals from over 100 institutions, and funded 12 projects from incredible institutions like the University of Pennsylvania, Stanford, Harvard, and others. And these are therapies, and I should say, even from our own backyard here, a project here itself at university hospitals as well as Case Western. So I think that we want you to know that we are on the leading edge of research into this area. And of course, we need your help to continue these research efforts. Please think about us. Go to uhgiving.org to make a contribution. You can earmark that to research. You can earmark it to healthcare workers. Your funds go a long way in propelling research. We want to thank you today for tuning in. Bob, it is always an honor and a privilege to listen to you as my teacher of infectious disease. We were kidding before we went on air. The lower levels of the class went into plumbing, as we called it, cardiology, and it was only the smartest who went into infectious disease. So, Bob, I'm just an interventional cardiology plumber, but I'm learning from you every day about the incredible complexity of emerging pathogens in global health. I want to thank everybody for tuning in. Bob and I enjoy these sessions. We enjoy preparing them, and we will see you soon, hopefully in the next 4 to 6 weeks, with an update on more vaccines and new therapies. Thank you very much.
Thank you.